Hello students, today we are going to study about fertilization and implantation. Today's topic is about fertilization and implantation. Fertilization is the union of haploid sperm with a haploid ovum to form fertilized egg or diploid zygote. The sperms deposited in the female reproductive tract undergo capacitation, which is a biochemical event that enables the sperm to penetrate and fertilize the egg. All these events, biochemical events which are taking place, that process only is called as capacitation. The hyaluronidase, it is a proteolytic enzyme which is present in the acrosome of the sperm that helps for penetrating the ovum. So once the head of the sperm it enters into the ovum, the nucleus of the sperm and the ovum, they unite to form fertilization. Once fertilization is over, the cortical granules which are present in the cytoplasm of the ovum, that it forms a fertilization membrane to prevent polyspermy. Fertilization by more than one sperm is called as polyspermy. The diploid zygote, it undergoes cleavage, repeated cell division, and that stage is called as cleavage to form ball of cells called as morula. At this stage, the embryo that it consists of fluid-filled hollow ball of 100 cells. And this stage of the embryo is blastocyst. The embryo that it consists of flattened single trophoblast and inner cell mass, rounded cells or inner cell mass, which is embedded in the endometrium of the uterus. This process is called as implantation. If the fertilized ovum is implanted outside the uterus, it is called as ectopic pregnancy. Ectopic pregnancy. The growth of the embryo may cause bleeding, internal bleeding, rupture of the fallopian tube, leads to, sometimes it causes death of the individual. Rupture of the fallopian tube, and sometimes that it leads to death. Twins are two offsprings which are produced in the same pregnancy as twins. Monozygotic twins, when a single fertilized egg splits into two during first cleavage, develop into twins of the same sex and share the same genes, or monozygotic twins, otherwise called as identical twins. Dizygotic twins or fraternal twins, when two separate eggs fertilized by two separate sperms, the twins are the same sex or different sex and they are non-identical. The third type of twins are Siamese twins. Twins are conjoined twins joined during birth. Students, you can refer the picture human fetus within the uterus. The fetus is covered by extraembryonic membranes, amnion filled with the amniotic fluid. The next membrane is, the outer membrane is chorion, and the uterus, this is uterus, and you can see the umbilical cord which is connecting the fetus with the mother, and placenta with the placentic villi. Allantois for removal of the excretory waste. This is only fallopian tube and the amniotic fluid inside the amnion. embryonic development. The blastocyst, which is transformed into 3-layered gastrula by the movement of blastomeres. The embryo, it is protected by 3 extraembryonic membranes. The amnion, filled with the amniotic fluid, that it protects the embryo from desiccation, from mechanical injury, and it is filled with the embryonic fluid that it regulates the temperature of the fetus. The yolk sac forms a part of the gut and forms the blood cells, earlier blood cells and blood vessels. The allantois is the structural base of the umbilical cord. The chorion, which helps in forming the placenta, that is the outermost membrane. The chorion, that it sends the chorionic villi into the uterus, forming the placenta, nourishing the fetus. Placenta is a temporary endocrine organ for nutritive, respirative, excretory functions of the embryo fulfilled. From the 3 germ layers, central nervous system, skin, mammary glands formed from ectoderm, kidney, bones, muscles, gonads from mesoderm, digestive and the respiratory tract, which is formed from endoderm. Placenta produces the hormones like gonadotrophin, estrogen, progesterone and relaxin hormones, which are produced by placenta. The next topic is parturition and lactation. The descent of the fetus causes dilation of the cervix of the uterus and vagina, that it results in fetal ejection reflex, which initiates the secretion of oxytocin from pituitary gland, and relaxin leads to the expulsion of the baby as partition, or it is otherwise called as childbirth, parturition or childbirth. The prolactin from pituitary and oxytocin, that it helps in lactogenesis. Colostrum, a nutrient-rich fluid filled produced by the human female immediately after giving birth, is loaded with immune, growth and tissue repair factors. It acts as a natural antimicrobial agent with all natural benefits, should definitely fed to the baby after birth. It contains more of proteins, vitamins, vitamin A especially, minerals, and rich in IgA antibodies against bacterial infection. The next topic is reproductive health. Amniocentesis is a prenatal technique to detect chromosomal abnormalities in the fetus, and this technique is misused to see the sex of the fetus. Birth control methods. There are 3 methods: natural method or rhythm method, barrier method and permanent control methods. Natural method or rhythm method, preventing the sperm and the ovum meeting together, natural method. Barrier method, chemical barrier using tablets, jellies or creams to inactivate the sperm. Mechanical barrier using diaphragms, cervical caps, wallets for preventing the sperm and the ovum meeting together. Hormonal barrier, pills to prevent ovulation. The pills are made of synthetic estrogen or progesterone. Intrauterine devices also can be inserted into the reproductive tract of the female. The third method is permanent birth control method, surgical sterilization method, tubectomy, surgical sterilization women. The fallopian tubes are cut and it is tied up, preventing the meeting of the sperm with the ovum. Tubectomy. Vasectomy is surgical sterilization in males. Cutting the vas deferens, preventing the sperm conducted to the urethra. So these two methods are permanent birth control methods. Next is sexually transmitted diseases, like they are otherwise called as venereal diseases like hepatitis B, HIV, gonorrhea, syphilis, candidiasis. All these diseases are sexually transmitted diseases. Next is about cervical cancer. This is caused by sexually transmitted virus called papilloma virus, and it causes abnormal growth of the cervical cells. Cervical cancer. Next is about infertility. Infertility is defined as inability to conceive even after unprotected sexual cohabitation. What are the causes for infertility? The causes they are referred as pelvic inflammatory diseases, uterine fibroids, endometriosis. The second factor is low body fat or anorexia in women, underdeveloped testes and ovaries. Female develop antibodies against her partner's sperm. Males develop autoimmune response to their own sperm. When we study about infertility, you must know a type of syndrome which is called as Mayer-Rokitansky syndrome. Mayer-Rokitansky syndrome. All women are born with the ovaries, but some do not have functional uterus, and this type of syndrome is called as Mayer-Rokitansky syndrome. Next, in this reproductive technology, all these are the techniques which are adapted. The first technique is intrauterine insemination. It is a procedure to treat infertile men with a low sperm count. Sperm collected from healthy donor introduced into woman's uterus by a catheter after stimulating the ovaries to produce more ova. This results in normal pregnancy. The second technique is in vitro fertilization, or it is otherwise called as test tube baby. Sperms and the eggs are allowed to unite outside the body in a lab. One or more fertilized eggs may be introduced into woman's uterus, where they may be implanted in the uterine wall and develop. Excess embryos can be preserved, cryopreserved for future use. IVF is used to treat many cases of infertility. The basic steps in IVF are ovarian stimulation, egg retrieval, fertilization, embryo culture and embryo transfer. 10,000 to 1 lakh motile sperms are needed for each egg. The zygote is allowed to divide to form 8-celled blastomeres, and then they are transferred to the uterus. This technique only it is called as embryo transfer technique. The next technique is cryopreservation or freezing of embryo is used to, there are more embryos than needed for single IVF transfer. Embryo cryopreservation can provide an additional opportunity for pregnancy through a frozen embryo transfer without undergoing another ovarian stimulation and retrieval. Next topic is detection of fetal disorder during early pregnancy. Fetal disorder can be diagnosed with the help of ultrasound scanning. The second method is amniocentesis. This picture that it shows the amniocentesis and the fetoscope. That is the third technique. In this picture, you can see that the fetus is surrounded by the amniotic fluid in the amnion. With the help of the needle, a small incision is made in the abdomen and a small quantity of amniotic fluid is sucked. It is taken and it is tested for the cells which are shed from the fetus. For any chromosomal abnormalities in the fetus can be diagnosed. Next technique is fetoscope. Fetoscope is used to monitor the fetal heart rate and functions during the late pregnancy. The average fetal heart rate is between 120 to 160 beats per minute. Students, today we completed the topics reproduction, embryonic development, reproductive health, reproductive technology and fetal disorder. All these topics are given in the textbook, pages 23 to 43. You can refer the textbook. So with this, we are completing the first unit. Thank you, students.